So, welcome everybody to our third session on FCS Express 7. For anyone that's joining us today for the first time, welcome. For anyone that's joining us and has been with us for some of these sessions already, thank you for joining us again. So just a brief introduction. My name is Kathy Daniels. I work as the manager of the core facility at Memorial Sloan Kettering Cancer Center, flow cytometry core facility. You'll also see here Sean Burke from DeNovo software. And he has been kind enough to go over with us multiple topics in FCS Express 7. And today for our third session, what we're going to be doing is we're going to be going over high parameter data analysis, including t-SNE and generating t-SNE plots, okay? This is t-distributed stochastic neighbor embedding for anyone that's not familiar with what t-SNE itself actually means. And Sean's going to go over how to generate these plots, analyzing and understanding, clustering, and of course there's always more that we learn as we go through these sessions, but that is the general overview for what we're going to be discussing today. So some important links before we get started. There's DeNovosoftware.com. So if you don't have FCS Express, you're interested in getting it, please go ahead and visit DeNovosoftware.com. There's also the FCCF website for Memorial Sloan Kettering, as well as our Twitter, which our handle is @flow MSKCC. We provide educational content. We share articles that we think are relevant for people in the flow cytometry community. We post parody videos, right? And some entertainment that we're trying to keep everyone entertained with during the current work from home scenario. So feel free to check us out there. Then we also have a link to ISAC and specifically Cyto University. So Cyto University is currently free for people working from home given the current situation. So we thank ISAC for that wonderful initiative. And then if you're looking to do any analysis in flow cytometry and you might not have any FCS files yourself to play around with, please feel free to visit flowrepository.org, especially for the topic we're discussing today with high parameter and t-SNE. What you can do is you can go ahead and download FCS files, and there are a good amount on flowrepository.org that are high parameter, and you can use that to play around with this high parameter analysis. Okay, some more important links. This is the cytometry Part A journal that you'll find a lot of information on. There's also the metroflow.org website. So Metroflow is the regional New York, New Jersey flow cytometry users group. And then lastly, if you saw that initial plot where we turned text into an FCS file, I just think it's really cool and we use that. So we have a little tutorial here from David Gravano on the YouTube. Okay. So these are the contacts for today. We have Sean Burke, again, from DeNovo Software, and this is his contact if you need to reach out to him. There's Rui Gardner, who's the head of the facility at Memorial Sloan Kettering Cancer Center with Gardner R at mskcc.org. And then there's myself, the manager of the facility, Daniel K1 at mskcc.org. So if you have any questions, we like to highly encourage everyone throughout the virtual classroom. This is really supposed to be interactive. If you have any questions, to please utilize the chat function in the Zoom's program. So please use that if you have anything that seems confusing, you're unsure, you have any specific questions, and myself and Rui will be monitoring that. And if we can answer it, we will. Or if we feel like Sean should answer the question, we'll interrupt him at the appropriate time to do so. So please utilize that. But if there are any questions after, go ahead and please realize you have these contacts. Okay. So with that, I am going to stop my screen share and pass it along to Sean, who we thank very much for doing these sessions for us.
Yeah, thanks, Kathy, and thanks for having me in here again today. And it's good to see a lot of familiar names in the attendee or participant list there. And do you see the screen coming through over here?
Yep, we're good.
All right, great. So let me make this a little bit bigger, and we can start talking about some of the high dimensional data reduction basics, including working with t-SNE. So again, thanks to the folks at Memorial Sloan Kettering for putting together this series, so we can learn a little bit more about how to use these tools and some of the other tools in FCS Express. And today's focus is going to be kind of a practical approach for understanding how to perform and interpret t-SNE plots with FCS Express. And I find one of the kind of best places to start with this sort of talk, instead of just going directly into the data analysis, is to cover some of the basics of what t-SNE plots are, what t-SNE kind of does at a high level. I don't want to get too much into the details of the mathematics and the algorithms. One reason, because I don't fully understand everything that's going on. And another reason, I think it might bore you a little bit, right? But what I do want to do is kind of relay a lot of information about what we've heard from our customers and researchers in the field about kind of common problems and misconceptions that they run into when they're performing or trying to perform t-SNE. So I'm going to kind of cover on a lot of that as well. Essentially what not to do. Learning how to do it in FCS Express is fairly straightforward. But learning about what some of those pitfalls are as you perform t-SNE is also kind of important. So where I want to start today is, why are we using high dimensional data reduction tools? Why are we even considering using t-SNE, and why? So really the problem is too much data, right? Having more parameters and more data is a good thing in my opinion. But essentially what you can end up with is, if you have a 20, 30 color parameter data set, essentially if you wanted to look at all of those parameters, side by side and compare everything, you could put in a kind of grid of data like this, n by n plot, so to say, where you look at every parameter versus every parameter. But really in the end, as a human, that's going to be really hard to distill, and really a lot of the information that's in those n by n plots isn't really useful. There's a lot of kind of phenotypic combinations that don't even apply to the experiment that we want to look at. So essentially, there's a number of solutions that can be applied for working with these types of data sets. So one solution is what I already mentioned, is kind of looking at 2 dimensions at a time and drawing manual gates around all of these different plots, and trying to use our own intuition and expertise to interpret what that actually means. Now, another way of doing that is doing really complex math in some sort of n-dimensional space, get out an answer, and then just trust that answer. And that's not an invalid approach, but generally you need some sort of statistician, somebody who's really good at programming, somebody who can put together that really complex math to get you the type of answer that you need. But there's another option, and that option is trying to find a way to visualize this really big data set on a single plot, and then really just trying to figure out how to interpret and use that plot to get the answers that you need. And really that's what t-SNE is. t-SNE is a way to get really high dimensional data into a plot that has 2 parameters, t-SNE X and t-SNE Y, and then trying to figure out ways to interpret that plot. So I like to show this little guy here, or this little model, right? His head is kind of blown from looking at all that data. So hopefully by the end of today, we can put that head back together and understand a little bit of this. But also, some of the things I want to talk about with dimensionality reduction. I already mentioned these algorithms and these tools. They kind of take this really high dimensional space and they distill it down into 2 dimensions, right? That's a low dimensional data set, but it's got to represent the original high dimensional data set in some sort of way. Now, I think the important thing to keep in mind is that generally when you're working with tools like t-SNE, these are exploratory data analysis techniques. They're not giving you any sort of empirical information about, these are the phenotypes that you're interested in, here's the cluster of that. They're not making any sort of determination of what you're actually looking at on that plot. But what they do is they simplify the data and make it easier to kind of search for patterns, which might not be easily observable by eye, especially if you're looking at n by n plots with 20 versus 20 parameters. They allow you to kind of get an understanding of the underlying fine structure of the data, and it allows you to look for natural separation of the data into groups. And that's something that humans are really good at, is trying to figure out, looking at some sort of image and trying to figure out how to distill that down into different little groups. So dimensionality reduction can, kind of come about a few ways. I have this little flow chart here. One of them is just using feature selection, and again, that's kind of using M by M plots. And then feature extraction is what t-SNE falls under. You can kind of follow this down here, where you create new parameters based on the data to get that out. And there's a lot of different forms of this. You may have heard of principal component analysis, things like isomap. There's a lot of kind of different names and algorithms for doing that. But let's talk about t-SNE in a way. So what t-SNE does in the end is, say you have a three-dimensional data set, and in this case, this is kind of a classic example of what's called a Swiss roll, where you're looking at parameters A, B, and C, X, Y, Z, whatever you want to call it, and you have 3 dimensions worth of data here, right? And what t-SNE essentially does is it performs a mathematical algorithm on this. It finds a way to kind of focus and highlight and exaggerate local structures, local populations, clusters, and then it takes that information and it represents it back on a 2-dimensional plot. And again, what you generally see with these are, this plot is called t-SNE X versus t-SNE Y. So this is kind of a t-SNE representation of this little Swiss roll model, right? And again, this is 3 dimensions. Now, when you're working with flow data, you might have 4, 5, 50 dimensions, depending on how many parameters you've acquired in your data set, how many parameters you're running through the algorithm. Of course, and showing a 20-dimensional data set in a Swiss roll is not so ideal. But really, that's the kind of idea. It's going to take this high-dimensional data, however many parameters you have, and map it into some sort of 2-dimensional space. So we can go in and we can say, all right, well, here's all the things that were read. Here's where they kind of appeared in 3-dimensional space, and we can kind of make some sort of representation of that. Now, I also like to start talking about, what do t-SNE clusters mean? And you see this picture up here. It's a bunch of little clip art images of dogs. And believe it or not, t-SNE is not limited to flow cytometry data analysis. In fact, it was around before. People were using it for different things. But this is a kind of fun example of taking some image-based data and running it through a t-SNE algorithm and see how it kind of clusters things. So in this little t-SNE clustering of these clip art images, you can see that the t-SNE algorithm took these kind of white dogs and put them up here. It took these black dogs and put them over here. There's some kind of brown dogs, or some dogs with colorful hair and longer hair. There's some kind of outlier dogs over here, right? And really what t-SNE clusters mean is up to you as the scientist, right? Or for me as the observer, I'm coming in, I'm calling these white dogs, black dogs, colorful dogs, brown dogs, right? And the same thing goes when we work with flow cytometry data, is that you have to look at this data and you have to assign it some sort of phenotype. The algorithm itself isn't some sort of magic bullet that's going to come in and say, all right, this is the phenotype and it's called white dogs, right? It's just going to put clusters and pieces of information in some sort of way that you can kind of distill it and try and label it. And you can kind of take this in a little bit more extreme example. I found this kind of image on the web here from, it looks like some folks at Stanford were clustering a bunch of images from the internet. And if we kind of zoom in on these 2 red regions that I have highlighted here, you can see that, what was found is that you have a little cluster of pumpkins here. I know this one's kind of hard to see. It's people fishing, or pictures of fishes. There's pictures of pies. There's pictures of mushrooms. But again, if you're looking at this from a scientific perspective, you have to come in here and label this stuff, right? So these guys are fishing. These are animals. And another important kind of concept with t-SNE is, just because animals is next to the fishing phenotype, it doesn't mean that these clusters are in any way related. And I think this is kind of a good example. I mean, it's pictures of people fishing. Fish are animals. Okay, that's a good idea. I guess they're kind of related. But very, close, you have pumpkins over here, right? Does that mean that pumpkins are related to fishing? Does that mean they're related to animals? We have some, what I labeled here, round foods. They're pies, maybe they're pumpkin pies, I don't know. But down here, there's mushrooms. So again, what you need to do as a scientist is, not just look at this and say, these clusters in my t-SNE charts are close to each other, so they must have some sort of meaning. In fact, just because something is close to another cluster when you're looking at t-SNE charts, it doesn't have any sort of meaning in that space. There's no sort of linking between phenotypes just because clusters are close to each other in space. There are other algorithms that do give some sort of relation. When you use tools like SPADE, it gives some sort of relation between the phenotypes. So things that are closer to each other in a SPADE tree are actually phenotypically more similar in a lot of ways. But don't get fooled. When you look at a t-SNE chart, just because things are close together, it doesn't mean that they're phenotypically related. So how do we interpret a t-SNE chart? You can see down here, I've got 3 plots. You can see here, I've got some kind of clusters. I've got some areas here that are in blue, some areas that are in red. And what happens with t-SNE charts is you're generally looking at these in some sort of density-based display with a heat assigned to that particular plot. And when you're looking at the heat is usually representative of the intensity of a marker. So in this case, you can see in the plot title here, it's labeled CD3. This plot is labeled CD4, and this plot is labeled CD8. So when we look at the heat here, we're seeing that this population here, this cluster, whatever we want to call it, it's hot for CD3, it's not very hot for CD4, and it's hot for CD8. So really what that means to us is that by comparing multiple plots and showing the heat for different markers, we can set gates on these to identify the phenotype. So if we come in here and we say this is our CD3 positive, CD8 positive population, we can come down here, we can say, all right, well, these are CD3, CD4 positive, probably CD8 negative, and kind of take that a bit further. And again, these are CD3 positive, CD4 negative, CD8 negative. And then you can take those gates, the gates that you create on some sort of t-SNE plot, and you can apply them to other plots to do downstream analysis. So, going back to, any of your kind of markers of interest, you can take these gates, you can apply, you can look for percentage of gated, you can look at MFIs, you can compare against different samples to see if sample 1 has a higher percentage or, higher MFI of CD3 than sample 2. But generally, that's how we are going to be interpreting these plots and how we're going to be looking at them. And when you have, say 20 markers, you're probably going to be looking at, maybe 20 of these t-SNE plots kind of side by side. And there's a lot of tools in FCS Express that we're going to show you today to kind of figure out how to distill those things a little bit more. Now, another thing about t-SNE clusters and t-SNE algorithms and t-SNE plots in general is you can see up here I have 3 plots, the CD3, CD4, and CD8, and I ran a t-SNE algorithm on these. And I have the same kind of plots down here, CD3, CD4, and CD8. And the question here is, what is the difference between these? And what the difference between these plots is how the t-SNE algorithm was initialized. So what that means is when you run a t-SNE algorithm, it has to start at some value. It has to start at some cell in the data set. So generally when you acquire data, you actually label cells. The first cell that came past the laser and gets acquired, that's cell number 1. And then cell number 100,000, if you stopped, it's cell number 100,000. Now, if you change where you start that t-SNE algorithm running, whether it be cell number 1, 5, 50, 5,000, the shapes of these plots and the way that they're distributed are going to look different. But if I came into this plot down here and I set a gate around this t-SNE cluster of CD3 positives, I can tell you just because I know looking at this data set, it's going to be about the same % positive as this one where it was randomly initialized a little bit different. So what that means is just choosing where to start can change the shape and the size and the location of a t-SNE cluster. So keep that in mind because really the size and the location of these clusters doesn't have a lot of meaning with t-SNE, especially when you kind of look at the global distribution of these things, right? So I know that this little cluster here, it's probably the same little cluster here, but just because it appears in the upper right doesn't give it some sort of, special information as opposed to it appearing in the middle of the chart. And it's something to keep in mind when you look at t-SNE charts and you go to conferences and you look at publications. Don't get fooled and say, all right, well, this cluster looks like it's pretty close to this one, so they must be phenotypically related. This one is nice and compact, and this one over here is kind of spread out a little bit more, so they must be different. It's just the way that the algorithm has run, and it's represented this high dimensional data set in two-dimensional space. Again, as I mentioned, there's other tools that are out there that do give you some sort of phenotypic information based on where the clusters are and how they're found. But again, we're going to be focusing on t-SNE today. And in that case, it doesn't have a lot of information. So again, these are kind of important points and misconceptions. These are things that I hear, when I go out to labs and I speak to people, things that we hear all the time when people write us into our support. The location and the size with t-SNE graphs doesn't have a lot of meaning for interpreting them. The clusters just contain groups of data that are similar to each other in some sort of way. And in the end, it's up to you as the researcher to identify and determine what those clusters are, and then ultimately to determine the biological meaning and significance of them, just like you would with standard two-dimensional plots. Now, I also want to mention, running t-SNE or v-SNE, or even if you add op-SNE into this, they're computationally heavy algorithms to run. It means that it can take a long time when you click the run button to run a t-SNE. And different software packages run t-SNE differently. You may have heard of local versus cloud computing. A good example of this is cloud computing is using something like Cytobank. Is there a question that came in? Nope. Oh,
No, I think it might have just been unmute on accident.
Cool. So cloud computing is something similar to Cytobank. You send your data to the Cytobank server, and then the computations get done there. Local computing is using your local computer. Today I'm going to be using the little laptop that I'm running on. And there's also the idea of working with t-SNE algorithms via R versus a native implementation. And R means we're using an R script, we're using the R software package to run that algorithm. And native means we're using the computer package, the software package that we're running in to run that. So just so you understand how this works in FCS Express, we work off of local computing. So we're going to use the resources in your local machine. We're going to run the algorithm natively, which means we're going to run it within FCS Express, and we're not going to rely on R to do so. And there's information out there. We have information on our website. Some people have done some comparisons, and this is actually getting a little old at this point. It's things kind of happen fast. This was done maybe 6 or 8 months ago before the latest version of Flojo came out. So keep it in mind, up here, time being high is a bad thing. It's taking a long time to run that algorithm. This is the sample size for a t-SNE data set that these kind of time comparisons were made. But you can see that FCS Express pretty consistently runs very quickly, even though you're running on the local machine, even though you're using the native implementation of it. FCS Express is one of the fastest tools on the market for working on your local machine to get this sort of information done. And when we look at this example of R and Flojo, the reason that these are kind of correlating is at least one of the previous versions of Flojo was dependent on R to run t-SNE. So of course, you'd imagine they run at the same speed. And the kind of timing differences that I could talk about here is, when you're using some other software, you might have to go out and get lunch. When you click run on the t-SNE button. In FCS Express, maybe you get a cup of coffee, right? Or from the other software, you're getting a cup of coffee. In FCS Express, maybe your result is already done, a few seconds after you click the button. So keep in mind, if you're using other software packages, FCS Express is generally very, very fast at doing these things, even using the laptop that you have in front of you right now. Now, there's little tricks to help these algorithms run more quickly. So you may have heard of things is acronyms like t-SNE and v-SNE and op-SNE, and they all do, essentially a very similar calculation, but in a little bit of a different way. Things, when you run a t-SNE, it's actually running this algorithm on all of the events in the data set. It's not making any sort of estimations about where events are and how they fall, so it can actually be a little bit slow. People invented a different type of way of running that algorithm called v-SNE, which uses something called the Barnes-Hut implementation, which we're going to see in FCS Express today, which kind of makes certain assumptions about the data, and it allows you to kind of sample less data points and then upsample that data later to more data points. So if I have a 1,000,000 event data file, if I run a t-SNE, I'm essentially going to run this algorithm on all 1,000,000 events. But if I run something like a t-SNE or a Barnes-Hut approximation, I can say run the algorithm on 50,000 events and make some sort of estimation for the other 950,000 events in that data set. And as you can imagine, the algorithm will run more quickly because we're actually sampling less data, but we are making estimations. And op-SNE is another way that we can kind of optimize these algorithms to run a little bit more quickly. The op-SNE comes from Anna Belkina's group at, I believe, MIT, or I'm sorry, Boston University. I don't want to get those confused. And there's some information, of course, on the internet about all the different kind of ins and outs of these algorithms. But I didn't mention upsampling and downsampling. It's something important that we should talk about and you should understand before running any sort of t-SNEs, or just running data analysis algorithms in general. Because by using tools called downsampling and upsampling, you can make these algorithms run a lot more quickly. So downsampling essentially, or subsampling, is a way of reducing the number of data points, so you don't have to run these algorithms on everything. But there are limitations to that. The trick is to keep as few data points as possible without losing the overall structure of your data. So if you have 1,000,000 data points, there's no way that we're going to downsample to 2 events, 2 cells in that data set and have some sort of representative information about all 1,000,000 events, right? So that's kind of overdoing it. That's over downsampling. But what you have to do is kind of find that sweet spot of how many events do I need to run to have an accurate representation of my data set overall? And there's no real empirical answer to that. I get the question all the time, well, how many events should I run in a t-SNE? It's really, you have to look at your data and you have to say, all right, well, if I ran it with 5,000 events, I seem to be losing some of these phenotypes. If I run it with 50,000, I seem to be getting all the information that I need. If I run it with 500,000, well, I'm still getting the information that I need, but the algorithm's taking 3 hours to run, right? So you need to kind of find that sweet spot. Now, when you downsample, you can imagine there's different ways to do this, right? To remove points from your data sets. One of the simplest ways of downsampling is called interval sampling. And that means, if I have 100 events and I take every 4th event, I take an interval of 4, then I can break that down 25 times, right? So I'll end up, if I take an interval of 4 for a 100 event data set, I'm going to end up sampling 25 events from that data set. So that's literally saying, it takes event number 1, event number 4, event number 8, event number 12, and do that throughout the entire data set. Simple random sampling is kind of another way of doing this, and instead of just taking a set interval, you kind of take a random sampling throughout the data set. And another type of sampling is called density-dependent downsampling. Now, it's kind of important to understand because we have some interesting problems that come up with flow cytometry data sets, or biological data sets in particular. Because generally, when we are looking at biological data, a lot of times the things we're most interested in are the rarest events, right? So we're looking at some sort of stem cell or minimal residual disease. The percentage of those events might be 0.1% or even 1%. So what would happen is if you do interval sampling or you do random sampling, what you get is kind of down here, you see our red population, our blue, our green, and our light blue. Well, the light blue is, not so dense. There's not a lot of events. So if we randomly sample 1,000 events from that data set, at some point we're actually going to sample out all of those light blue events. And if those are our super interesting events, our 0.1% or 1% of cells, well, you're going to get rid of all of them, and then you're not going to be even able to analyze them, right? So density dependent sampling is something that you can do to avoid that. And essentially what it does is you can set target densities and weighted densities, and you can say, all right, well, take less events from things that are very dense, like this blue population, take more points from less dense areas, and then when we run our algorithm, run it on that subsampling of the data. And FCS Express has kind of one of the unique abilities to say for t-SNE, you can do simple random sampling, you can do interval sampling, you can do density dependent downsampling. So again, there's a lot of ways that you can do that. And what you have to do at that point is, all right, well, I've taken only 50,000 events of my 100,000 event data set. Well, what are we going to do with those other 50,000 events that we didn't run the algorithm on? And that's called upsampling. When we examine the cells that were not modeled, or sorry, not modeled, but were not included in the algorithm, what we can do is kind of assign those events to what we define as the closest cluster or the nearest neighbor. And we make an estimation about where those events should fall in the t-SNE X and Y data sets. So when we do our upsampling, we can view the statistics for the entire data set of say 100,000 data points instead of just the 50,000 that we downsampled to. But again, we're making estimates, when we do that. And the trade-off is, again, if instead of having to wait, 3 hours for a t-SNE algorithm to run, maybe we can wait 5 minutes, right, and get our information a little bit more quickly. So I do want to kind of show you, we're going to jump into FCS Express in just a second here, but here's an example of upsampling in FCS Express. We have a sample data set on the left here that was downsampled. And if we don't apply upsampling to that data set, you can see that we actually did a interval sampling of every 3 events. And what this looks like at the data level, when you're looking at the raw data in FCS Express, is that in a data set that it was not upsampled, we only end up with t-SNE X and Y coordinates for cell 1, for cell 4, for cell 7, for cell 11, and everything in between is kind of not sampled, and it's not accessible to us with this t-SNE X and Y coordinates. So again, we're looking at 3,000 events here. That's how many events we downsampled to. But if we upsample that same data set, we end up with the t-SNE X and Y coordinates again for cells number 1, 4, 7, 11, but we fill out everything in between with some sort of best guess, some sort of estimate about where those events should fall, even though they weren't sampled. And one of the things that you'll find is that when you're looking at things like density plots, even 2-dimensional plots, these plots are going to look a little bit nicer because they just have more events, right? This has 11,000 events in it instead of 3,000, and we're also able, when we set gates, to relate the information from those t-SNE plots back to the full data set and not just some of the downsampled data. Now, another consideration that I hear all the time, and this is one of those problems that folks write to us in our support all the time, is that you have to compare apples to apples. And what that means is if you want to compare multiple data files using t-SNE, you cannot run a t-SNE on sample 1 and then run the same t-SNE on sample 2, and then compare the t-SNE charts for sample 1 and sample 2 side by side. In that case, you're kind of comparing apples to oranges. Really what you have to do is merge all of the data files into one data set before you run the algorithm. And once you do that, you can run the algorithm, and then you can deconvolve the data sets down the line into their individual samples for further analysis. So the question that always comes up is, isn't that going to take a long time to run my analysis? The answer is yes, right? So every once in a while, I get somebody coming to me and they're saying, all right, well, we have a control set of 20 patients, a experimental set of 40 patients, and then I have something in between. If you want to analyze that 60 patient data set, you do have to merge all 60 data files. You have to run the t-SNE algorithm on them, and then you have to deconvolve that later. And we're going to show you how to do that in FCS Express. But yes, if they're all 1 million data points and they contain 28 parameters, it's going to be a really big data set, right? But there are reasons, why you should run t-SNE on that. But again, just as a quick example, here's a t-SNE run on one sample. This is sample 1 up here on the top. We have another sample, sample 2 on the bottom. And you can see that if we run the same exact t-SNE on these, the t-SNE plots look different. They're kind of differently shaped. The clusters have different groups of clusters in them. And you can't compare these side by side. It's comparing apples to oranges. So what you need to do is merge those data files. And when you merge that, you're going to end up with a new t-SNE plot. And again, this merged data set won't look anything like sample 1 or sample 2. It's going to have its own t-SNE x and y coordinates. And what we do with that is we create a separate plot before we're ready to analyze our t-SNE data sets with what's called a file identifier parameter. And what we can do is with file 1, we can set a gate around that. With file 2, we can set a gate around that. And then we take that gate and apply it to the t-SNE chart. And then we can compare file 1 in one t-SNE plot to file 2. And then we can get an apples to apples comparison about what, say in this case, the CD3, we can see that there's a lot more CD3 positives here, or, a tighter group of cells here in file 2 than in file 1. And of course, you can take that downstream and say, all right, here's our CD4s from file 1, here's our CD4s for file 2. So again, it's just a matter of deconvolving those data sets later on. So with that background information, you're probably saying, oh, this seems really hard. Well, why do people do it? Well, one reason is you can reduce the number of gates that you create, right? By looking at multiple t-SNE charts side by side, you can set one gate and you can say, all right, that's my CD3, CD4, CD8, CD19 positive population, because I can look at one set of charts and set one gate rather than having to, create a nested hierarchy of many gates to do that. Also, kind of one of the dreams of this is finding populations that we missed or couldn't otherwise see in a big set of N by N plots. So looking for, samples that respond to a drug treatment, looking for rare populations, looking for hard to gate phenotypes. These are some other kind of valid reasons to do it. But honestly, one thing I see all the time is because it looks cool, right? You go to a conference, you put up a bunch of t-SNE charts on your poster in your presentation. It looks really nice. I mean, these charts are really beautiful looking. I can't tell you how many times I've gone to a conference and have seen t-SNE charts on a poster, and they could have done the same analysis with 2x2 plots or histograms or something like that, but again, it looked cool and it probably helped get it published that day. So I don't want to discount it, right? Looking cool is certainly one thing, but don't go overly overboard for trying to create t-SNE plots just because somebody says you need a t-SNE plot in here, right? If you're finding the same sort of information that you would derive from a t-SNE plot with a standard type of flow cytometry analysis, I don't see any sort of reason why you would have to go and use a t-SNE plot. So I know that's a lot of kind of preamble to showing how this works in FCS Express, but those again, I think are the really common and important things that I've found that a lot of our customers have been coming to us asking questions about, again, kind of common pitfalls. I don't want to see you, working in FCS Express, doing a whole bunch of t-SNE analysis, and then writing us and saying, well, why do these t-SNE charts look different? Because you didn't merge all of your data files together, right? And then you have to go back and do it all over again. So try and keep those things in mind. But go ahead.
Sorry, Sean, just really quickly, I just want to make sure that people remember that they can use the chat function and utilize that if they have any questions as we get into this portion of the talk as well.
Great. Yeah, keep questions coming in if you have them. So what we're going to do now is show you how to create a t-SNE in FCS Express. And you're probably saying, oh, you spent so much time talking about this other stuff. Well, it's really easy to do it in FCS Express. So that's why I save it here for last. But essentially what I'm going to do is I'm going to load in a data file in our data list. And as I mentioned, if you're going to run a t-SNE, and especially if you have multiple data files, you're going to want to merge data files together. Now, the way that we do this in FCS Express is when you go to load data files, you choose the Merge FCS File option, and you multiple select all the data files that you want to run your t-SNE on, or you want to merge together. This is also useful for other things. If you're, some people call this concatenation. It's the same thing, merging data files. You can get this data, you can drag it into the layout, you can insert some sort of plot for this data, And what we'll be able to do is change to any of the parameters on our plot, say forward versus side scatter, and we can create some sort of gate on this. And I'm going to just call this cells. And I'm going to drag this gate out. And what I'm going to show you is that we also have that file identifier parameter, right? So the first file that we merged together is file 1. The second file that we merged together is called file 2. And if you're using what we call plate heat maps in FCS Express, you can actually insert a little plate heat map for this, and it will show you, whatever keyword, sample identifier. In this case, I chose to put the sample ID in here, so I can actually visualize this a number of different ways, whether I want to use two-dimensional plots or gate heat maps. They both work kind of similar ways to deconvolve this data later on. So I'm going to put this plot over here. I'm going to put my little plot here. I'll just look at CD4 versus CD8 because I can. And one downsampling that we've already done to this data before we even knew it is we created a gate, right? So we created this gate called cells, and all that this gate does is it removes debris and it removes our bead population. So just right off the bat, we're doing downsampling. You do it all the time. You just don't even know it. We call it gating, right? And if we want to go in and run a t-SNE on this data set, we're going to go into the tools. We're going to click on transformations. And in the little drop down, there's a number of different transformations you can run on this data set, one of which is t-SNE. So we'll create a t-SNE transformation. And you can see that this is already automatically populated with some stuff, right? So one of the first options it's going to ask you is, when you run this t-SNE, do you want to run it on a subset of cells? It's going to say, do you want to apply a gate? So we do. We want to apply the cells gate because we don't want to cluster all of the information from our beads in here because it's probably not useful for the biology of this experiment. So, whether you have one gate or a bunch of gates, maybe you have, singlets and live dead staining, you can, use whatever gating that you want before and apply that gating before you even run a t-SNE. Now, when you,
Oh, sorry, Sean. I just wanted to pop in there too and say that, the inclusion of the viability in the singlets is really important to make sure that you don't have the artifacts as well. Because since dead cells or antibodies can non-specifically bind on those dead cells, and aggregates will have that as well. So that's going to be important in your analysis.
Yeah, exactly. And, the other consideration is, if you've got 10,000 events that are debris or doublets and stuff like that, there's no reason you want to use the computing capability of running t-SNE to run it on things that you're not going to be analyzing, right? So the more events that you can remove to achieve, whatever type of experimental result that you want, to remove extraneous pieces of information or extraneous events from the data set, the better, right? So in this case, I actually downsampled to 70% of my events. I already got rid of 30% of the data, 30% of that is beads, and now I don't have to use my computational power on my machine to run it on that extra 30%. Now, when you run a t-SNE, you can pick and choose which parameters you want to include. I'm going to choose all of these in here. I'm going to run the t-SNE and look for clusters amongst all of these populations or all of these parameters. And then we get down to our sampling options. So again, this is another downsampling step here. The first one was just creating a gate. But if I want to say in this t-SNE, use 3,000 events to run this t-SNE on, that's what this sample size actually means. It means run the t-SNE on 3,000 events. Now, we set a default here of 3,000. That's not some sort of empirical, way of setting this. We literally just chose a random point to say this is going to be our default, right? The default could be 3,000, it could be 50,000, it could be 500,000. It's just a default value, and it's up to you to kind of come in and define where you want to set your sample size. But if you just want to kind of see how things run, you could probably leave it at 3,000. It will probably run pretty quickly the first iteration that you run this. And we do have different downsampling methods, which I discussed earlier in the slides. So if you do want to investigate things like target densities and density-dependent downsampling, you can actually use these for t-SNE plots. Now, I also mentioned earlier that you could run exact t-SNE, and that's going to run essentially ignore sample size, and it's going to run the t-SNE on everything. Or you can use the Barnes-Hutt approximation, which is very similar to v-SNE. And the Barnes-Hutt approximation, again, is going to kind of do just that. It's going to make an approximation. It's going to make some estimates and assumptions about the data. And these parameters that are here allow you to kind of change variables about how those approximations are made. So how much approximation to make, how many times to actually run this algorithm. There's something here called perplexity that we'll come back to in a few minutes. But what will happen here is when I want to run a t-SNE on something, what I do is I drag and drop that t-SNE algorithm from the transformations onto our plot. Once this plot says transformed in the title, or once I'm able to kind of click on the screen again, that's how we're going to know that that transformation is actually complete. The other way you'll know it is complete is because you're now going to have a t-SNE X and a t-SNE Y parameter. So we can pick and choose and switch over to that. Now, the other thing I didn't do here is I didn't estimate t-SNE for unsampled events. And essentially what that is the upsampling step that I mentioned before. So in this case, the plot looks kind of sparse because I didn't upsample. I'm only looking at 3,000 events here. But if I click on estimate t-SNE for unsampled events, what's going to happen is the t-SNE is going to recalculate, and it's going to calculate kind of nearest neighbors and best estimates for the rest of the data in that data set. And once it's done calculating, you're going to see that this updates and that we have more events here. Now, another way that you can kind of get t-SNE to run more quickly is using this use op-SNE button. If I choose use op-SNE, it kind of hard codes a few different parameters to the algorithm. Again, I don't want to go over the mathematics of it in a ton of detail today, but op-SNE is a way essentially that you can kind of speed up running t-SNE, making kind of, I guess, certain assumptions about the data sets. But again, you can visit our website. There's a lot of information on the differences between Barnes-Hutt and v-SNE and t-SNE and op-SNE. I would say go to the published literature to kind of look at that.
Sean, just to interrupt you very quickly, we have 2 questions that came through, and I think now will be a good time before move forward. So one question was about merging of the FCS files, and they asked if you could merge non-FCS files, such as MQD files that might be from another instrument. That was the first one.
Yeah, so MQD files would need to be merged a little bit differently. In the interest of time for this talk today, I'd say, please contact me directly. Write me at sean.burke@DeNovoSoftware.com, or reach out to our support team at support@DeNovoSoftware.com. There's other ways of merging this data. The merge FCS file option will only work for FCS files, but there are tools in our data tab here for essentially loading up and then batch merging out data sets like MQD. So FCS Express is a little bit different than other software. We can read those MQD files directly. So do keep that in mind. You don't have to re-export to FCS files. But if you want to use the merge FCS file function just from the data list, you could export. If you want to merge them in a little bit more roundabout of a way because you want to maintain MQD file format, we can probably accomplish that through using these tools up here.
Okay. And then, thank you, Sean. And then the second question before you get going is someone had asked if you downsample based on a parameter, should you include that channel in the t-SNE calculations?
I'm not quite sure I understand the question, because when you downsample, you don't downsample on a parameter, you downsample on the entire data set. So this isn't saying take 3,000 events from the CD8 parameter, it's saying take 3,000 events from the entire data set. But again, it's a conversation that we can follow up on. I'm sure it's just a matter of figuring out how to phrase that question. But again, you can write me, you can write us at support@DeNovoSoftware.com as a bit of a follow-up, and we can also follow up after the call if you'd like.
Perfect. Thank you, Sean.
Great. So we've got our t-SNE X and Y here, but we don't know what's kind of being displayed on this chart, right? It looks like everything is kind of evenly distributed. I'm actually going to get rid of my t-SNE navigator here. I don't really need it anymore. And what you need to do to kind of start working with this is formatting this plot. So one thing I want to show on this plot, which I can do by right-clicking and formatting, I can double-click on the plot. There's a number of ways to call this up. But when I switch over to looking at a legend, I can make a legend visible, and you can see that a legend now appears on the plot. And these legends are interactive. So if I want to say base the heat on this plot for CD3, well, I change it to CD3, and now I'm looking at the heat of CD3 on my t-SNE X and Y plot. And essentially what you can do is if you want to look at, say, t-SNE heat of CD3 against CD4, you can copy and paste, you can create a new chart, and then we can kind of start looking at this and say, all right, that's our CD3 positive, it's also CD4 positive, that's our 3 and 4 CD positive population. But we do know you're going to have a lot of these parameters, so doing these one by one is not going to be ideal. What we have is if you have a density plot shown with t-SNE X and Y in FCS Express, you can click on the multi-plot tab and you can say color by all parameters. And this is a nice tool because you can say, all right, well, if I wanted to look at the heat of CD3, CD4, and CD8 on density plots, I can do that. It's going to create a few t-SNE plots. It's going to group them together. And again, what I can do is I can say, all right, well, let's show the legend. And instead of squishing it over to the right, I'm going to put it on the bottom. And then just like that, I'm able to look at the heat of CD3, CD4, and CD8, right? They're all kind of nicely grouped, they're displayed, and I can come in here and start doing some gating and other things I need to accomplish. So again, you can do that with as many of the different parameters that you have in your data set, and you can start working with them in that way. So the other thing I'm probably going to want to do here is if I want to compare, my CD3, CD4, and CD8, but I know I have two samples, is I'm going to want to start using some of these file identifier parameters. So I'm going to look at the file identifier versus, say side scatter. It doesn't matter which parameter you look against here. I'll change this back to counts. And what I can do is I can set up a gate and I can say, all right, well, this is file 1. All right, so here's file 1, and I'll create another gate, and I'll call this file 2. Let me bring this back over here so you can see it. And now I have two gates, file 1 versus file 2. And if I want to compare the t-SNE populations of file 1 versus file 2, well, I'm going to need two sets of t-SNE plots. And I can say that for this set, apply the file 1 gate. And for this set, apply the file 2 gate. And what I'm doing now in these two sets of plots is I'm comparing file 1 versus file 2. Now, I do see here that it's kind of difficult to visualize what's going on with my dots here. So I am going to format these plots a little bit differently. I'm going to go into one of my options here, and I'm going to say use a little bit less resolution, which is going to kind of make my populations appear a little bit more nicely. And that way I'm going to be able to kind of visualize these. Now, if you want to, I know you're not just going to have two files a lot of times. If you have many files, these gates, you can group multiple populations together or multiple files. You can see here as I extend this gate, which is the file 2 gate, it's what's applied over here, that it's encompassing, multiple data files. So if I have a group of 3 controls and 6 experimentals, just set the gate, encompassing all the controls that you want. And the other kind of nice thing that you're going to see is that as you move gates around, all this information updates in real time. So you can imagine if I have 6 data files and I don't want to look at 6 plots side by side, I can kind of move this gate from file 1 to file 2, and I can kind of visualize how those changes to the gates or the different data files is happening in real time. Now, it's also a nice way to kind of normalize how many events are being shown on a t-SNE plot, because we do understand sometimes you collect, say, 50,000 events from file 1 and 100,000 events from file 2. Well, if you use something like maybe the time parameter over here, let me recreate my gates for file 1 and file 2. just give me a second to do that. So that's my file 1. This is my file 2. So if we kind of need to normalize how many events are coming into these different things here, essentially what I can do is adjust that gate, right? You can see that if I want to include, maybe about 37% of events for file 1 and the same number of events for file 2, essentially I can adjust that gate and include as many events as I want to help normalize between the two different data files. So once you have all of this done, you're probably going to be setting some gates on your t-SNE plots. So, doing that, it's just as easy as, setting gates anywhere else. I'm just going to call this t-SNE gate. And when I have that t-SNE gate created, essentially what I can do is start investigating all of the other parameters in this data set in our standard two-dimensional plots. So if I want to look at, say, CD4 versus CD8, and I want to get that t-SNE gate applied, what I can do here is as I adjust this t-SNE gate, I can see on that plot what those populations actually look like, right? So I can see that this is a CD8 positive, CD4 negative. I can see down here that this is some sort of CD4 positive, CD8 negative. But we do understand, again, that you're going to have more than 2 parameters to look at a time. So, generally what folks do is they have, a lot of their kind of classical type of flow cytometry style plots in the analysis where they can kind of backgate and they can compare, what these different t-SNE populations mean. But another quick way of getting to that is if you insert a histogram, you can compare all of the different parameters in the data set using this histogram, using a new tool that's in FCS Express 7. If you right click on this and you say add parameter overlays, what you're going to be able to do is select a number of different parameters, say maybe CD4, CD8, CD19, get rid of that one. I'm going to say switch this to stacked. And what we're going to be able to do is quickly visualize across all the different parameters. Let me get my t-SNE gate applied so we remove that kind of outlier beads peak over there. And we can look at the MFI values across all of the different parameters that fall within this t-SNE gate very quickly and easily. So again, what I can do is I identify, all right, well, this is some sort of CD4 positive, it's some sort of CD3 intermediate, it's negative for 8, it's negative for 19. I can come over here, all right, there's our 8 positives, they're negative for everything else pretty much. And again, just like any of the plots in the software, you can make these look a little bit more beautiful. So I always do like when I'm looking at any sort of histogram overlay, maybe come in here and give it some smoothing, maybe give it a fill, maybe give it a transparency. And by doing so, again, it kind of beautifies that and makes it ready for publication. So when you do identify, the populations of interest that you want to look at, you have everything, ready to go for publication. So again, creating these t-SNEs and getting them applied to plots and working with the plots is not very difficult in FCS Express, right? I think it's more of a matter of kind of understanding, some of the pitfalls about, how to merge data files, how to change what you're looking at on the X and Y axis for these things, what type of plots to visualize these in. And once you do so, again, these are kind of helpful to not just replace the kind of standard analysis that you're doing. So you can see here that I have a kind of another data set. I've already put together some standard analysis and gating, but they can help you kind of visualize some of these data sets in a different way, right? So in this case, I've created a t-SNE. I've gone back, I've done some backgating to confirm what these populations are. And I can visualize, okay, here's my B cells, here's some monocytes, my RO, RA populations. And again, you can use this back and forth between your kind of standard flow cytometry data analysis and working with the high dimensional data reduction tools. So I think it's good to use these kind of in combination, not necessarily to replace one with the other. There are cases that you can do that. But, one of the really nice things about this is, again, if I have many different data files, and I merged them together and I've identified where the populations are, very easily at a glance, I can compare, say, file 1 on the left to file 2 on the right, and maybe there's a lot more NK cells in file 2, or there's a lot more NK cells in treated versus untreated, right? So I think that's one of the uh, big tools that people are trying to use t-SNE for, is to look at across many different parameters, across many different data files, comparing groups of samples to one another. And again, essentially, I think what we did today is give you a lot of the practical tools or the practical information for generating that sort of information with FCS Express. So I think that there was a few questions that probably came through there, Kathy. Is that correct?
Yeah, so you answered the other two earlier about the MQD, and then how when we do our downsampling, it's based on everything, right? And it's not specifically a parameter, right? So those questions were already answered. And we had one more about specifically the Barnes-Hut approximation. And they asked if does Barnes-Hut approximation perform additional downsampling to the one that you initially defined when you did the t-SNE analysis?
I'm going to have to double check, honestly, but I believe it does not. I believe the Barnes-Hut approximation is the downsampling that we choose here is what's being used for the Barnes-Hut. So if we say a sample size of 3,000, the Barnes-Hut approximation of the algorithm is being run on 3,000 events. It's making certain approximations based off of the variables that are here, and then once it's done, it's upsampling that back to the rest of the data set.
And then we have another question about the proximity of clusters. So they're asking if clusters in the t-SNE plots, are they not defined by proximity and only by backgating? Does proximity of clusters have no significance? Or is there a definition of a cluster based off of proximity, and what that cutoff might be as far as how close together those clusters
Yeah,
are?
it's a really great question, because kind of going back to the beginning of the presentation, I mentioned kind of over and over again that the clusters, just because they're close to each other, doesn't really mean anything, right? But what is close and what is far? I'm looking at this kind of cluster here. Well, maybe there's actually two clusters that are there. So generally when we talk about things in this kind of clustering space not having any meaning, it's generally kind of like global distances, right? So like this cluster is obviously very different from this one. It's in a different space altogether, so they're not at all related. But just in the same right, I mean, if this is a cluster here and this is a cluster here, just because they're this close to each other doesn't mean that they're related. But there are at some point it does become a localized cluster, right? An event that's right next to another event, of course, is part of that cluster. So generally what you'll be doing to kind of deconvolve that is through backgating, right? So again, you can go back and you can backgate the samples to kind of pick that out. And I think this other example I had here was a pretty good example. If I can find my RO and RA cells here, let's see if I can pull it up and show you. So you can actually see here that the way that we've clustered this, these red ones are t-SNE RO CD4s, and these blue ones are t-SNE RA CD4s, right? So this whole cluster is T cells, but somewhere in between there, there's RA T cells and there's RO T cells. And the way that we identified where this breakpoint at is not in the t-SNE chart. It's not because of, these were clustered differently in the t-SNE chart. Essentially what we did is we went back and we looked at our backgating of RO and RA, and we said, all right, well, we took a gate from our RA positives, applied it to the t-SNE chart, looked where they fell in this population, and then set a gate on our t-SNE plot to indicate that those were RO cells. And the same thing, we looked at our RA gate from whatever plot that we set it up on, we applied it to our t-SNE plot, we set a definition there and said, where are the RA cells? Now you might be wondering, you're like, oh, isn't that kind of like cheating, right? Well, what you would do is, all right, so you've defined that on some subset of your data, but imagine you had 100 data files that you're comparing side by side, right? So when you do that, to set the gates on the t-SNE chart, what that allows you to do is then when you're visualizing these across many groups of samples or across from one sample to another, then you're using the t-SNE chart to very easily in one plot say, all right, here's our CD4s, here's our ROs, and here's our RAs, rather than using 2 or 3 plots to distill that information. So again, you kind of use these back and forth, right? Some cases people just use t-SNE charts, but more often than not, you're going to be using t-SNE charts, you're going to be using kind of classical type of flow cytometry data analysis, and using them side by side to get the benefits of each of them, right? So again, kind of coming back to it, looking at this, I'd never be able to look at this plot and say, all right, that's RAs and that's ROs, but I could probably, look at this and say, all right, that's my CD4s altogether, but I need backgating to go in and further define where there's kind of subpopulations within a major cluster itself. So hopefully that wasn't too long-winded of an answer for
No,
that.
no, not at all. The more information we have, the better. So does anyone else have any more questions for Sean on t-SNE in FCS Express 7, or we're all good? Give him a couple of seconds.
I'd also like to say, if you do have questions, reach out to us at support@denovosoftware.com. I've got this up here. So I know, questions are going to come up when you go back and you start doing this, right? Don't hesitate to reach out to us with any questions. We're always happy to help you out there.
Yeah, wonderful. All right, so thank you to everyone for attending today. And next week, we're going to be at the same time going over, I believe, cell cycle and proliferation for our next week session at the same time. So we welcome all questions. If you have to reach out at our emails that we provided. I'll also be sending out a PDF of the slides from the introduction, and we hope everyone stays safe. And thank you again endlessly to Sean for doing this for us. All right, thanks everyone.
Thank you. Take care. Bye-bye.
Bye.